So we're really interested in how cells can come together and reform a tissue, and how we can encode information in the extracellular space, the space around cells, to get them to do that. How is this information encoded? Well, cells and tissues are normally in a extracellular matrix, which is proteins such as collagen, carbohydrates, and fats that the cells secrete and assemble around themselves to form a tissue.
So as Adam mentioned, the corneal endothelium does not regenerate, and disease or injury leads to corneal blindness. And it's estimated that about 10 million people suffer from corneal blindness worldwide, and a lot of that is due to damage to the corneal endothelium. So my project focuses on engineering freestanding protein matrices to regenerate the corneal endothelium. So our project is different from current approaches in the literature in that we're trying to actually characterize the mechanical, structural, and compositional properties of the corneal endothelium and its membrane, where a lot of other groups are using tissues and proteins that are not native to the corneal endothelium.
So one of the really unique aspects of what we're trying to do is, unlike maybe traditional tissue engineering approaches that will use some sort of synthetic polymer to build a tissue engineering scaffold, we're trying to understand how cells build with individual proteins and make their own scaffold. And so we're really trying to mimic that process. And right now we're using something called surface-initiated assembly of proteins to do that. It's very similar to the way cells assemble proteins on their surface, and we call that a biomimetic approach. It's kind of a bottom-up way of rebuilding a tissue. So to understand how the cells in the corneal endothelium build the endothelium, we're actually looking at the structure of the endothelium in both the cow eye and the human eye to understand what that structure and material is like, and what we need to do to mimic it. And she's looking at various aspects of this endothelium, including the mechanical properties, as well as the structural and chemical properties. And one way we have to mimic that is something called soft lithography. It's really an interesting technique. So we actually create small rubber stamps with features on here smaller than the eye can even see. These are micro and nanoscale features. But what they do is they replicate the structures we see in the actual corneal endothelium. And then we can engineer those into a substrate or a surface that we can then grow cells on and expand them and build a new bioengineered endothelium.
So the instrument that you've been seeing behind us is a laser scanning confocal microscope, and it allows us to image the 3D environment of the corneal endothelium. So what we have here is an image of a bovine corneal endothelium intact. And what we see here is that the cells have a rather hexagonal shape, which is very characteristic of the endothelium. In blue, we have the nucleus of the cell. In red, we have ZO1 protein, which is the protein that binds the cells tightly together and controls the pumping of water and nutrients across the corneal endothelium. If we switch over and look at how these cells look cultured on glass, you can see that they have a very irregular morphology with a lot of stress fibers, barely any of the tight junction protein, and much fewer cells. So what we can do is optimize the stiffness and protein coating of surfaces to bioengineer a corneal endothelium that looks much more like this natural corneal endothelium. So here we have a corneal endothelial layer that we have cultured in the lab by matching the mechanical and stiffness of the surface with that of the native corneal endothelium and the extracellular matrix proteins. We were able to create a layer of cells that looks much more like the natural corneal endothelium than the glass surface that you saw here. So here we have a 3D image of the surface that I had showed you that we cultured the corneal endothelial cells on. So you can see the types of images we're able to capture on our confocal laser microscope. So we still have the cell nucleus and then that ZO1 protein that shows the tight binding and the cell shape of the hexagonal character of these cells. And as we move it around, we can look at different side views and the top view of the image that we collected.